Okay, so we're going to study aerobic respiration today. So if you just want to sit back and listen and watch, that's fine. Or if you're into taking notes while I do that, that's okay too. We're going to just start on this blackboard right here. And let's just kind of review what we already did. All right, let's just make sure we know where in the whole story we are. So cell respiration, you already know, can be anaerobic or aerobic. You know that. And we've already done the anaerobic talk. Right? And today we're doing aerobic. I'm going to fill in this part over here. So let's review a little bit. What is the first step of both of these things? The first step of anaerobic or aerobic, whichever type. What does the cell do first with the glucose? Yes, thank you. Glycolysis is the answer. Yes, glycolysis. Oops. Yes, okay, glycolysis. And where in the cell does glycolysis happen? What part of the cell does glycolysis? In the cytoplasm. Excellent. Good. In the cytoplasm. There. Okay. So whether you have oxygen or not, whether you have a mitochondria or not, you are doing glycolysis first, and that can occur in the cytoplasm of any living cell, whether it's one of your animal muscle cells or whether it's a plant cell, a mushroom cell, an amoeba cell, a bacteria cell, no matter what kind of cell it is, every living cell has those enzymes that can grab onto glucose and start breaking it down, floating around in the cytoplasm. That part of the metabolic reactions has been conserved since day one, the very first tiny little bacteria 3.8 billion years ago that figured out how to do glycolysis, that passed it down in its DNA. There are genes for all of the enzymes that run glycolysis in the cytoplasm. And that has been conserved in our evolutionary history. Okay, now let's go back to anaerobic respiration. What's the second step? Once glycolysis has broken glucose down into pyruvates, what's the second step of anaerobic respiration? What do you have to do? Remember to recycle those NAD pluses to keep glycolysis going. And that's called, and it's something that we do to make wine, and it's something that we do to make beer, something that is helpful in bread making. Fermentation, yes, that's it. Fermentation, which also happens in the cytoplasm, by the way. I can't spell cytoplasm today, apparently. Okay, there we go. Fermentation in the cytoplasm. And let's review how many ATP do we get per glucose if we're only doing anaerobic respiration? Oh, yes. So we have 2 ATP per glucose, right? Per glucose. Not terribly efficient, but if you are a tiny little yeast cell or bacteria cell, that can keep you going, and you can do that. Even our skeletal muscle cells will do lactic acid fermentation for a little while, but they can't do it for very long. And you know how sore your muscles get when you do that. All right, now let's go back over here. All right, second step of aerobic respiration. Now we're in a mitochondria and we have oxygen. What's the second step? Remember, it was named after a German. His first name was actually Hans. Yes, the Krebs cycle. Good. All right, so the Krebs cycle happens next. Good. Very good. All right. And that has to have a mitochondria. So you need to, for this, need to be eukaryotic to be able to figure this second step out. The final step of aerobic respiration, that really is the biggest payoff of the whole thing, is the electron transport chain. Yes, the ETC, electron transport chain, also in the mitochondria. So you have to be a eukaryote in order to do that. All right. Final ATP yield per glucose of aerobic respiration. What do we have this time? Those mighty mitochondria, how many ATP are they churning out per glucose molecule? More than 2. 36 is actually the generalized number. Generally, you're going to have between, the textbooks say between 34 and 38, depending on the type of cell and depending on other little details that you don't need to worry about. Okay, but generally the magic number is 36, and that is per glucose molecule. Okay. All right, so much more efficient. So kind of like if anaerobic respiration was music, it would be like James Taylor, laid back country music stuff that, you know, oh, we'll get some ATP out when we're, you know, when we get there and all that. Aerobic respiration would be like AC/DC, Back in Black. Can you think of another one? Give me some music for aerobic respiration. What are we talking here? High energy music that you guys really like. What would you say? Come on, guys. Come on. Something fun. No? I know Rachmaninoff versus Debussy, maybe? All right. Well, we'll just forget about that and keep going. Okay. So I'm going to erase this and start another set of notes here. All right. The other thing that you really need to know is how redox reactions fit into all of this. So if you guys remember your chemistry, you remember that acronym REO-GR, maybe? Or maybe you learned oil rig, but I probably won't. Oil rig. Yeah, you remember those, right? Okay. And what did those help you remember? They helped you remember what's happening with the electrons in oxidation versus reduction, right? So what does that mean? It means that oxidation means molecule is losing electrons, right? So REO-GR, lose electrons, oxidation. And reduction means that the molecule is gaining electrons. And this is important in this biology lecture because there are some very, very important redox reactions happening, which if they were not happening, you would not be making all that ATP in the mitochondria. Making energy using a mitochondrion by the Krebs cycle and the electron transport chain is really all about oxidation and reduction reactions, about the flow of electrons. So let me explain to you how exactly that's going to work. So there are two very important players here that I need to introduce to you. They are called electron carriers. And their names are NAD+ and FAD. These molecules are called electron carriers. And what does an electron carrier do? It carries electrons. How simple is that? Okay, so what you need to know, though, is what the molecules look like when they're either not carrying electrons or when they are. That winds up being important. So when NAD+ has electrons added to it in a reduction reaction, it becomes NADH. There, okay. So this is the oxidized version of that electron carrier. If you add electrons, but it becomes reduced. So NADH is carrying high energy electrons. That makes this a reduction reaction. Okay, so NAD plus electrons gives you NADH. That's a reduction reaction. And that happens a lot in the flowchart that I'm about to show you of aerobic respiration. The other electron carrier, FAD, is going to be a very similar looking thing. If you have FAD and you add electrons to it, it becomes FADH2. That is also a reduction reaction. Now, it turns out that reduction reactions generally can't happen unless there's an oxidation reaction happening nearby. So these reactions are coupled. That's why they're often called redox reactions. You can't have electrons being added to something without electrons having been taken off something else. So the something else is the glucose being broken down and bonds broken. Every time you break glucose down into smaller and smaller and smaller bits, you are releasing electrons from the covalent bonds that used to hold it together. You're oxidizing glucose, and you're reducing these electron carriers. So it looks like this. You have glucose at the top of this big basic flowchart, right? And little by little, you are breaking it down into smaller and smaller pieces by glycolysis and by the cycle. Those two things are breaking glucose into smaller pieces so that glucose by glycolysis, which is the first part of this, is breaking that 6-carbon molecule into 3 carbon molecules, right? And when that happened, when you broke those bonds and you created 2 smaller molecules, you released electrons when you did that. So this oxidation reaction got coupled with the reduction reaction of NAD plus to NADH. So the electrons that came off of here, when the bonds were broken, get added onto this guy so that you now have an electron-rich carrier. This keeps happening. It keeps happening again and again all along this pathway, right? Because when the Krebs cycle comes along, you get to the mitochondria, the 3-carbon molecule then becomes a 2-carbon molecule. And then eventually you get a whole bunch of 1-carbon molecules. And at the end, you have carbon dioxide, 6 of them, and 6, you also have 6 water molecules, which is all that's left of that original glucose molecule. And all along the way, you kept breaking the molecule down and creating NADHs. And eventually, you also create one of these. There's only one spot where you make the FADH2s, right? But all of those guys, all of the electron carriers, when they are carrying those electrons that came off of the glucose as it got oxidized little by little by little, they now are going to go somewhere and do something useful with those electrons that they are carrying. All right, so remember, all of these reactions here are oxidation reactions. The glucose broken into 3-carbon molecule, broken into a 2-carbon molecule, and then finally broken into single carbon molecules. All of those reactions are oxidation reactions. And as those oxidation reactions happen, they release electrons, which get picked up to reduce the NADHs and the FADH2s. Now this is important. This stuff down here at the bottom, the carbon dioxide and water, this is waste. It's absolutely useless to the cell. It's the product of aerobic respiration, but this is all just waste. It has no energy value. These are waste molecules. The cells are going to get rid of them. They might keep the water. The water is always useful, but the carbon dioxide, you need to get that out of there because that's not conducive to anything that a cell needs to do. The money maker here is this stuff. These electron transport molecules are going to pull their electrons where? Where are they taking the electrons? Where would be the obvious place to take electrons in a mitochondria? We've done the Krebs cycle. What's the last step? Yes, they are taking it to the electron transport chain. Electron transport chain. Yes, well named because it's not working. Because the electron transport chain, what does it do? It transports electrons, and it needs to get those electrons from these guys. All these guys are taking the electrons to the electron transport chain, which is going to use the flow of those high energy electrons, right? All those high energy electrons that are being carried over there, I'm trying to make negative signs there, are using the flow of electrons to make lots of ATP. That's where the flow of energy is really going here. It's a flow of electrons. The electrons are coming off of glucose and the food molecules and being put onto the electron carrier molecules. The electron carriers carry the electrons to the electron transport chain. The electron transport chain has a way of using the moving electrons to fuel the making of ATP, sticking phosphates onto ADPs. And we'll see how they do that in a minute. But then there's one final thing that we kind of need to mention. This is called aerobic respiration, isn't it? And yet we have not yet once talked about oxygen. What is oxygen doing in all of this? It doesn't seem like it pops up anywhere. It doesn't seem like it's important at all. It turns out that oxygen is very, very important, but not until the very final step. This is what oxygen does. Oxygen gas is very electronegative. Oxygen atoms are very good at taking electrons, accepting electrons from molecules. They can take electrons even when the electrons have lost all of their energy. They can even accept low energy electrons. And by the time the electrons go through the electron transport chain, they've lost a lot of their energy. So oxygen comes in and takes the electrons out of the chain and gets them out of the way. So what do they do? And I'm going to need more explanation for you than that. But oxygen is the final electron acceptor. And that's not going to make sense until I really talk to you a little bit more about this, right? So oxygen is the final electron acceptor. Once the electron has gotten through the chain and they've helped finish making all that ATP, oxygen picks those electrons up when they've lost their energy and puts them together with neighboring hydrogen molecules to make water. Right? So the water that's here in the product is a result of oxygen coming in, taking those electrons and taking hydrogens that are always floating around and making water. Okay, that's a lot to digest. I want to go and take, oh yes, Julia.
I have a question. I maybe missed But like, where does the oxygen come from?
The oxygen is what you breathe in or what is in the environment around the cell that's doing this.
Oh, okay, thanks.
Yeah, no, that's a really great question. Is there, we kind of assume that you know that all these things are floating around and available for the cells, and it's not necessarily that something that you know, is it? But oxygen is going to be in the environment around the organism that needs to do this. And the nice thing about oxygen gas is that it is actually small and nonpolar, so it can diffuse right through phospholipid bilayers without needing any kind of transport proteins. So oxygen can make it all the way inside mitochondria without having to use any transporters or channel proteins to do it. But it's a really good question. Oxygen, as long as it's around, as long as it's either in the atmosphere or in the water around the organism, and as long as it's diffusing in, then it can do this. Then it can provide this service and be the final electron acceptor. When I get back from break, we're going to give you a break here, okay? When I get back, I'll go into more specifics about how exactly it does that. But you guys know, if you run out of oxygen, you're not long for this world, right? And actually, this is the reason why. It doesn't look at all like oxygen is doing anything that important. Final electron acceptor, so what? Well, because it's so connected to this, to the making of those 36 ATP for glucose, can't happen unless oxygen comes in and kind of gets electrons out of the way so that this process can keep going. And that's going to make more sense after the break when we talk some more. Okay, so right now, I want you to go take a break. I'm just going to turn